Hello everyone, myself Rana Kumbar Singh. Welcome back to Grand Archive classes. I have asked you some questions in my previous lecture. Let's see the answer of that questions. Okay, first question was genetic engineering start with the discovery of which enzyme? Answer is restriction enzyme. Second question was that which enzyme lead to the joining of DNA segment? It was ligase. Third question was that difference between exonucleus and endonucleus. Exonucleus, these nucleus cleaves base pair of DNA at their terminal end. Second point is that act on single strand of DNA. Okay. Now see endonucleus. They cleave DNA at specific point except the terminal end. Okay. They cleave one strand or both strand or double strand DNA. It is now clear. Now fourth question was difference between plasmid DNA and chromosomal DNA. In plasmid DNA are circular extra chromosomal DNA. First point. Second point was that not associated with histone protein. Third point was that replicate independently. In chromosomal DNA generally linear form associated with histone protein, and last point is that replicate with genome only. Okay, clear. Now fifth question was what is an gel electrophoresis? See, the fragment of DNA obtained by cutting a DNA using restriction enzyme and is separated by technique called gel electrophoresis. It is a short answer for 1 marks question. If a question come in 5 marks, you have to explain with figure. Okay. Now see, sixth question was what is palindrome? These are the group of letter. They form the same word when read both forward and backward. See example Malayalam. It is now clear. First point restriction enzymes we have completed. Now second point cloning vector. What is a cloning vector? A small piece of DNA that is maintained in the organisms and then inserted in a foreign DNA fragment. It is done for cloning purpose. Cloning means making a multiple copy of anything. Okay, see. What is a chromosome? It is basically thread-like structure found in the nucleus of animal and plant cells. It is basically made up of proteins and single molecule of DNA. Okay. Now see, plasmid and bacteriophage. It is used vector for cloning purpose. Now what is a vector? It is basically easy linking of foreign DNA and selection of recombinant from the non-recombinant things. Okay, it is now clear. Bacteriophage. It is a virus that infect bacteria. We can say normally virus that infect bacteria. Okay, it is clear. Now see the following features. It is divided into 4 classes. First one is ORI, origin of replication. Second point is selectable marker. Third point is cloning site. Fourth point is vector for cloning gene in plant and animals. First, see origin of replication. The sequence from replication start or DNA will join to any fragment of sequence and start multiplying or copying in the host cell. It also controls the number of copy of the linking DNA. It is clear. Now see second point, selectable marker. What is an selectable marker? It is basically process of identifying a non-transformant. Now what is an non-transformant or transformants? It is basically introducing a DNA into the host cell. Okay, it is now clear. Now see, there are some antibiotics such as ampicillin, chloroamphenicol, which have basic role. Just like ampicillin used to treat bladder infection, pneumonia, gonorrhea, and this chloramphenol it is used in eye ointment to treat or conjunctive disease, plague cholera or typhoid fever. It is now clear. Now see this tetracycline. It is used in skin and lymph nodes. Fourth one is kanamycin. It is used in basically tuberculosis. Now see this cloning site. To link the foreign DNA, the vector need to have specific single recognition site for commonly used in restriction enzymes. See this E. coli vector PBR322 showing a restriction site. Sites are N2, CLA1, E. coli, PV1, PST1, PV2 and SAL and BAM. This is basically represent to link the foreign DNA to the vector. It is now clear. How does cloning site represent? Cloning simply means, by the name of the cloning, we can say that making a multiple copy of any gene. It is now clear. Now see fourth point, vectors for cloning gene in plant and animal. Okay, see Agrobacterium tumefaciens. It is basically pathogen of dicot plant. It normally that transform tDNA and transform normal plant cell to the tumor. Okay, it is now clear. From tDNA to tumor. Okay, now see in animals retrovirus is basically transform normal cell to the cancer cells. It is now clear. All the fourth point, ORI, selectable marker, cloning site, vector. Okay, now see, अगर आपको वीडियो ये पसंद आई हो प्लीज उसे आप लाइक करें, सब्सक्राइब करें और शेयर करें.